personalized approach to rare diseases is on the rise. John Crowley, chairman of the board and CEO of Amicus Therapeutics, sets the scene and hosts a conversation with Dr. Michael Yeaman, Dr. Joe Weimer, and Dr. Katherine.
In both kids, it's heart that's the real threat to their lives.
How much more time do we have?
Megan, maybe a year. Patrick, less.
I wish that we had a drug to treat Pompeii, but we simply don't. I'm sorry.
Dr. Stonehill.
Yeah.
This is John Crowley. All the researchers out there say that you're a genius on the verge of a scientific breakthrough.
I'm not on the verge of anything.
How much would it take to prove your theory?
The odds against you are crushing. But where does that leave your kids when the dad has no job and no health insurance?
You're right. This is crazy.
But I can't just sit around and wait for my kids to die. I promised him that we'd raise 500.
That's all?
1,000.
Is it 500 or 1,000?
500,000.
Are you totally insane?
Apparently.
Is he worth it? The Stonehill guy?
He's really eccentric, but his science is way ahead of everybody else's.
Do you have a wife?
Ex-wives. 2 of them.
Yeah? How come?
Because I'm so easy to get along with. Figure any dude in a business suit can help me raise venture capital and run the company. But who's going to be half as motivated as a dad who's trying to save his own kids?
Hi, everybody. Good day. I'm John Crowley, and it's my pleasure to host this session on a personalized approach to rare diseases. I am very excited about where we are today in the field of genetic medicine. I go back to the late 1990s when 2 of my children were diagnosed with a rare form of muscular dystrophy known as Pompe disease. And we were told then that there was little research, there was nothing that could be done, go home and enjoy the time that we had with our children. To us, that wasn't an acceptable answer, and it launched us into the world of medicine, into the field specifically of rare diseases and genetic medicine. And thankfully, with the intervention of a treatment, our children's lives were saved. But it also launched us into this field, realizing that with more than 7,000 rare diseases, there is so much unmet need. And we have to marry that against the promise of some tremendous science. In the last couple of decades, we've seen that science in small molecule precision medicine, in biologics, now increasingly in gene therapy, and someday soon the promise of delivering medicines with gene editing. So we're very excited about what the future holds, and particularly excited to be able to host this today with the Cura Foundation and with the Pontifical Council for Culture. I was reminded of some of the words of the Holy Father, of Pope Francis, back in 2019 on the World Day of the Sick, where Pope Francis described what he called a selfless love for every human being without distinction of language, culture, ethnicity, or religion. That's what we see in our world of genetic medicine. These diseases know no geographic boundaries. And a couple of themes that I'd like to see come out in our great panel ahead. One is the notion of access. We are developing great medicines increasingly. We also need to make sure that every person in need, everywhere in the world, has access to these new great medicines. That means more approvals. It means a commitment for companies and entrepreneurs like ourselves to make sure that our medicines are fairly priced and broadly accessible. And in those parts of the world where they can't afford these medicines, to make sure that we get them to patients in need. It also means that we need to continue to think about the regulatory pathways to realize that this is a battle against time as much as it is against the mistakes of nature. So ensuring access, but also another point I'd like to see to come out in this panel is continually to make sure that patients are at the center of everything we do and everything we think about. In developing medicines, it really is this virtuous circle. It begins with early stage research, oftentimes in research centers and universities. It moves on to entrepreneurs and small companies. It's partnered with venture capitalists, investment bankers, people to bring the capital necessary to do all this great research. Our hospital systems, our physicians, our nurses, regulators, people ensuring access again along this virtuous circle. And when we get to the top of the circle and we realize that we've made a medicine that's extended and enhanced life, very rarely is it a cure. And we need to do it again until that day where we truly have cures. But again, at the center of all of that really is the notion of the patient. And how do we consistently have that mindset that when we make decisions, we need to think, if you had this disease, whatever it may be, where you were the mom or a dad of a child, what decision would you make? That's, I think, the heart of being patient-centered, that mindset. And it's something I'm very excited about because at the end of the day, ultimately what we are delivering is hope to patients in need. And it has to be real hope, tempered with reality. But with the great science that we have today, I'm increasingly excited about being able to deliver on that hope in the decade again. So if I began with a quote for Pope Francis, I'll end with a quote that I think frames this conversation ahead well. Pope Francis said that only if we see ourselves not as a world apart, but in a fraternal relationship with others, can we develop a social practice of solidarity aimed at the common good. So with that, my hope is that we can advance the common good in this great discussion. So I'm pleased to kick this off and welcome our panelists. I am joined today by Dr. Michael Yeaman of the UCLA School of Medicine and the Guthy-Jackson Charitable Foundation. Also joined by my colleague, Dr. Jill Weimer, both of the Sanford Research Institute, as well as Senior Vice President at Amicus Therapeutics, and also by Dr. Kathy High, President of AskBio. So we're going to have this conversation. We're going to talk about rare diseases and talk about some of the great advancements that we're starting to see, building on years of research and experience, I know, for each of you. So Michael, let me begin with you. When you think about the study of rare diseases, paving the way for personalized and customized healthcare where no one size fits all, how do we think about it? How do you think about it?
John, thank you very much. It's great to be with everyone today. You know, our work with the Guthy-Jackson Charitable Foundation focuses on a rare disease called neuromyelitis optica, or NMO. And NMO occurs when the immune system misrecognizes normal proteins in the central nervous system as though they were foreign, reacts to them, leading to inflammation and loss of myelin, which in turn can lead to vision loss and loss of mobility and other issues. And what's so interesting about NMO is that even though the patients share a similar immune disorder, the disease can manifest in many different ways and can have vast differences in each patient journey. So in this respect, we appreciate each person, in each case, in and of themselves, and we always approach the solution as treating the person, not the disease. And so what we're really trying to do is find the best intersections between personalized medicine and precision medicine. And, you know, over the last decade of work, we've accelerated the science of NMO and advanced the clinical medicine and diagnostic accuracy. And we've gained credibility with industry and brought a bunch of expertise and perspectives together in a way that after 100 years of no approved therapy for NMO, in the last 2 years, we now have 3 approved therapies that again find the intersection between precision medicine and personalized medicine. So John, I think rare diseases really can open doors to many other solutions.
Now, Michael, that's a great point. I think, you know, people ask me from time to time, you know, why do you work in the rare diseases? You know, there's so few patients versus much more prevalent disorders. And part of what I share is that by unlocking the biologic secrets of understanding human genetic disease, we will unlock the secrets to much more prevalent challenges of human health. So in one hand, there are many rare diseases. Taken together, they're as common as all cancers combined, or more so. But then also when you start to unlock some of these biologic secrets, particularly in the neurologic field, you start to understand better diseases like Parkinson's, Alzheimer's. In some cases, a lot of the diseases of neurodegeneration really are just a series of human genetic diseases. I know in the CNS field, increasingly, children who, you know, maybe over the years were diagnosed in broad categories of epilepsy or cerebral palsy or even autism, increasingly are being genotyped and realized to have single gene or sometimes multiple gene mutations. So it is an exciting field in that regard. Jill, let me turn to you. A lot of opportunities to help people. Where do you think the greatest opportunity lies in using genetic targeting for rare diseases?
Absolutely. Thanks, John. I think, just building off of what Michael mentioned, over the last decade, last 5 years, last 2 years even, we've seen just a blossoming in the genetic medicine field. And I think a lot of this is built off of standing on the shoulders of giants such as Kathy and leaders in the gene therapy development field, and starting to recognize that it's not one size fits all approaches. I think many times I've heard it's simply as simple as copy and paste to restore a gene into a patient suffering from a rare disease. But we know that that's not the case, that each person has unique genetics, and even if they have mutations in a similar disease or gene, it can result in very different diseases. One of the examples I think that we're seeing are in diseases like frontal lobe dementia, where you have mutations in progranulin that can also lead to a pediatric disease, one of the forms of Batten disease. And so similar approaches can be used, but just looking at the unique genetics and what are the things that you need to be able to supply to that patient. So looking at personalized approaches, some of the pioneering work that's being done in antisense oligos to be able to customize those therapies to a unique patient's genetics, I think are really helping us turn the corner to developing personalized medicines, but also just not thinking about just the wild type gene. Are there things that we can do to enhance gene therapy approaches or genetic approaches to get better cellular targeting, to get better distribution within a cell, get it to the right places and get it turned on and functioning more efficiently? So I really think the next couple of years are going to be just remarkable to watch in how these technologies unfold in genetic medicine.
Great, thank you, Jill. And you're right, Kathy, you've been one of the great pioneers in this field. You have been. When you look today, most gene therapies fall into two categories where we've seen approvals, and there've been very few. Single gene disorders that you're familiar with, and then also in the CAR T cells for leukemia or lymphoma. Where do we see the next approvals?
Well, John, I think there continue to be exciting programs for other single gene disorders. I would think of the hemophilias. I would think of Duchenne muscular dystrophy and other muscular dystrophies as well. So I think that we will certainly see more approvals for single gene disorders. And, if you look at Zolgensma, for example, or Luxturna, these are AAV gene therapies that were approved on relatively small data sets, 40 or so patients for both of those. I think that what that tells you is that it testifies to the dramatic effect in a single gene disorder of putting the gene back into the right physiologically relevant target tissue. The clinical results are dramatic, they're clear-cut, and you don't need a very big trial. Most of the effort, in fact, comes around CMC, not around trying to figure out what you're seeing clinically. And I think that those kinds of dramatic results have fueled an interest in trying to develop gene-based strategies for complex acquired disorders. You mentioned a couple of those, Alzheimer's disease, Parkinson's disease, and so forth. And I believe that you're right, that what we're learning from the study of single gene disorders gives us lessons that are important to carry over into complex acquired disorders. And I do believe that there will be successful gene-based strategies for a number of those disorders. But I think in the near term, what we will continue to see are approvals for single gene disorders. But I'm excited about the future for a wide range of diseases.
Yeah, I think we all are. And, when you think about, you got in the United States, you and Jeff and the team at Spark oversaw the approval of the first gene therapy in the United States, then followed by Zolgensma for spinal muscular atrophy in 2019. And there haven't been any since. And you brought up an important point on CMC. So that's manufacturing. That's the analytical tools to make sure that we understand the quality and the quantity of gene therapy vectors that we're delivering. And it's a really important point too, that yes, that basic biology and science is very hard, but we make great progress. And when they work just profoundly. but we've got to get the manufacturing right. We've got to get these analytical tools right. In some respects, perhaps the entire field got a little ahead of ourselves. And I think together as an industry, we're working through with regulators, making sure that we've got the right quality of materials and have the tools to understand the quantity. In gene therapy, I think the process, the manufacturing process is the product. And we've got to get that right. So terrific points. Michael, maybe if I turn back to you, is there an opportunity to use platform technologies to treat many of these diseases, or do they all need to be, bespoke or custom tailored and developed one at a time? How do you see that?
Yeah, John, I think there are opportunities. There will be some new and remarkable opportunities to apply, let's say, the power of the immune system, for example, to solve many seemingly different diseases. For example, in NMO, we're using tolerization to reset the immune system in a way that takes an autoreactive disease and essentially tames the immune system so it no longer reacts to proteins that are in us that would otherwise be normal. And the point here is that we can use the power of the immune system to deal with any protein antigen that might be the cause of an autoimmune disease. So in that sense, it's sort of theme and variation. And this could apply to any autoimmune disease for which an autoantigen is known. And the other side of that same coin might also be true for other diseases that are related to immune dysfunction. And I'll just use the example of cancer, which we often think of as, cells gone rogue. And that part is true, but cancer emerges because the immune system fails to detect and destroy these cells. So even if the earliest possible detection, cancer cells have already figured out how to evade and outpace the immune system. And I think what we can do is use sort of inverse tolerization to prepare the immune system for cancer cells it has yet to see. And in that sense, prevent cancers that might have emerged otherwise.
Yeah, very exciting, Michael. Jill, let me ask you. There are a lot of promising advances in technologies for rare diseases, particularly in the gene therapies. What are you seeing in your work at Sanford or Amicus or the field more broadly?
Absolutely. I think one of the things for me personally that I've experienced over the last 5 or 6 years is as scientists, we're so often trained that we have to go it alone, that we have to come up and conceptualize the ideas and put those into practice. But I think if we can actually work in partnership and as teams, and teams for me has been working with family patient foundations, really helping them drive the direction that we're going and get input on drug development, partnership with industry. Really, I think we can move mountains. I think for so long we've tried to do it alone as individuals, and I think we need to look for unique ways that we can actually partner. And I'm not saying this just from the context of paying lip service to collaboration where maybe pharma sponsors a particular project in an academic lab. I think it's really getting the thought leaders together into a room, bouncing ideas off of each other. One of the things that I look back to in the neuroscience field from a decade ago was the BRAIN Initiative. And the BRAIN Initiative was not intended to fund neuroscientists like myself. It was actually intended to fund computer engineers, structural biologists, physicists to come into the neuroscience field, use their tools that were very unique from an individual neuroscientist trained to synergize and really move the brain mapping process forward. And I think in drug development for rare diseases, we can learn a lot from that process and really accelerate what we're doing by bringing in expertise from folks that maybe we wouldn't normally collaborate or partner with.
Great, thank you, Jill. Maybe Kathy, back to you. You highlighted a few moments ago one of the challenges that we're addressing as a field, and that's manufacturing. One of the other challenges is safety broadly. We need to make sure for these medicines, particularly in gene therapy, that they are both effective and safe. What do you think about the recent safety events in the field and how they limit development? Maybe your thoughts on technologies to address it.
Well, John, before I tackle that, I just want to echo Jill's comment. Drug development, and particularly gene therapy drug development, is the ultimate team sport. And it really puts a premium on communication of innovative ideas and just playing as part of a group. Nobody can do that sort of work alone. So I do want to put safety into context. I think that many people know that over the last year, we've learned as a field about a number of safety events across the board in gene therapy. Some in lentiviral mediated gene therapy in hematopoietic stem cells, some in AAV trials where the vector was infused either intravenously or given neurosurgically to the brain. And it's important to consider all these things in context. If you look at the number of new investigational new drug applications submitted to the FDA over the last 5 years in the field of gene therapy, in 2017, the number shot up pretty dramatically to over 100. The next year, it was over 200 new investigational new drug applications. And the following year, it was even more. So there's been a dramatic increase in the activity in the space. And so I think it's important to recognize that in the context of greatly increased activity in the space, many more trials, many more people enrolling in trials, that yes, it's not surprising that in that type of setting, we begin to learn the limits of what can be done with these vectors safely. But I also think that we're looking at some issues related to, for example, who can enroll in these studies. And can a person with liver disease expect to safely be injected with a vector into the liver? Or, so I think we're learning some limits about what we can do. I think that part of what we're seeing is simply a consequence of many more trials going on. The good part about that is that the pace of learning accelerates. And the challenging part is that we will be learning some limits.
I think that's very important. And also, too, to remember as we look at this, there is no cookie cutter approach. I think we have to look at each molecule and each disease and each patient population. And again, bring in that patient perspective. What risks are they willing to tolerate for a chance for a much improved and longer life? So that risk-benefit assessment lens is going to be important for all of us, whether it's developers, we as innovators, regulators, to think about and to continue to get that patient perspective. Michael, when you think about gene therapy broadly as a field, yes, we have come a long way to go. How do you see that fitting into the therapeutic arsenal, and in particular, how it may differ from other kinds of interventions that we're more traditionally used to?
Right, John. I think as Jill and Kathy have emphasized, we need to sort of blend the promise of sort of genetic modifications with the need for safety and targeted strategies. And so, for example, in immune-based disorders, including autoimmune disease, infection, cancer, transplant rejection, and others, there's usually more than one immune system component that is a little bit out of balance, either working too hard or not working hard enough. And gene therapy can be used to deliver what we call gain in function solutions, where we turn up the immune system a bit, for example, in cancer, or it can be used to deliver loss in function solutions, where we turn things down. And examples might be, for instance, in NMO, there in many patients is an extra set of copies of genes involved in the complement system. And you can imagine targeting some of those extra copies by either gene editing or deletion strategies that can potentially turn down the autoreactive status of the immune system. The same is very likely true for the genes involved in the molecular handshake between cells like T cells that govern the immune response and other cells that present antigens to them in a way that can diminish autoreactive responses.
Great, thank you, Michael. Jill, back to you. When this wonderful science, but ultimately we have to translate it into and through clinical studies. Right now, clinical studies are still very complex. How do we think about clinical trials for rare diseases, ideas for accelerating evaluation, reducing costs? What are your thoughts?
Yeah, I don't think I need to tell this panel that one of the caveats that we face in the rare disease space, particularly in pediatric rare diseases, is that if we follow the normal time course for a traditional clinical trial, many of the patients will have deteriorated and succumbed to the disease in the time that we get our ducks in a row and process through a phase 1, phase 2, and phase 3. And I think traditionally the time to complete that safety to efficacy to the phase 3 trial, there's the gaps in between, the processes to analyze that data to get approvals to move to the next steps. So one of the things that I think you're seeing more and more discussed in the rare disease space is this process of seamless clinical trials, where you would just evolve within your clinical trial to phase 1 to phase 2 to phase 3 without those gaps. Adaptive clinical trial design, where we're learning as we go along, right? We're racing as fast as we can to get these medicines to the patients as needed. And we're learning as we go along. So the ability to adapt our clinical trial design after it's initiated, built upon learnings that we experience as a part of that clinical trial. And that could be as simple as changing the primary outcomes, secondary outcomes, changing how we're analyzing the data as we're going through the process, I think is also going to lend to the rare disease space. And then as Michael had touched on, sort of these platform technologies, and looking to our comrades in the cancer space to look at trial designs like basket trials, where we might be looking at multiple different drug indications within one disease, or umbrella trials where we're looking at multiple different disease or genetic indications with the same platform. I think really is going to be the only way, John, to speak to how we accelerate these processes and how we cut costs to be able to move these drugs towards the market and towards patients.
No, those are great points, Jill. And I really think about it as this truly holistic system. If we can reduce the barriers to bringing more medicines to patients, we'll bring the cost down, we'll increase competition, we'll increase innovation. So again, in that notion of a virtuous circle, I think that's incredibly important. I would just add, as we think about the use of biomarkers in this studies, I think those will be increasingly important. Use of natural history studies. We all get calls from patients and family members on a daily basis, asking about what they can do in their diseases, oftentimes very, very rare diseases. And, what I usually tell people is try to help us find more people living with your child's or your disease so that we can help them when we do have therapeutics, but also so we can begin tracking the natural course of their disease to build natural history studies. Because in many of these diseases, it's just not acceptable to use placebo controls. You can't do it in diseases like Batten disease that we work in, or other rare, devastating disorders. So we have to be creative. And that also means thinking about biostatistics. Traditional biostatistics are likely not to apply in many of these therapeutic areas. So to think more creatively about using the adaptive studies you described, Jill, but also using tools like Bayesian statistics and multi-domain responder indexes, looking at the entire totality of the data in advance. And that's going to require all of us, again, to think about this in that benefit risk assessment. And again, against the notion of time. How do we beat time as much as we beat nature? It's a big question, Kathy, but I'll throw it to you to conclude. What do we think the future of gene therapy holds?
Well, John, I think that if we look at the history of other new classes of therapeutics, and I think about monoclonal antibodies, they were not products when I was in medical school. And I remember that there was a lot of controversy around issues about using monoclonal antibodies as therapies. The first one was approved in 1986, OKT3. We still use it in transplant. The next one was not approved until 1994, so a long gap. And then 3 years later, Rituxan was approved, along with another monoclonal antibody. And the year after that, 4 more were approved. So it took more than a decade. But by the end of that first decade, the number of products was really ramping up. And by the end of one more decade, monoclonal antibodies were 8 of the top 20 biotech drugs. I think that we're in a similar inflection point now for gene therapy. I think that with all of this activity in the space that I alluded to earlier, will all of those be products? No, probably not. But right now, gene therapies have a better percent likelihood of success than most other classes of therapeutics. So meaning that people, are really moving toward things where they have very strong proof of concept in animals. But I think that we will continue to see the pace of licensing, the pace of approvals of gene therapies increase. And so that's one thing that I think. And I think the other thing, gene therapy is really different from other classes of therapeutics. A person undergoes a fairly intensive period of medical management when the gene therapy is administered and in the initial weeks following that. But then the trade-off is that they attain a new and better status of health and well-being that allows them to move forward with far less medical intervention than they might have been accustomed to. And to me, that's very exciting.
It is exciting, again, continuing to move patients along that virtuous circle until there is a cure. And, I hope that gene therapy my hope is that, the days of a doctor telling parents that your child has a rare disease and I'm sorry, there's nothing that can be done, that's just not acceptable anymore. And I hope that the future brings a discussion where a doctor can say, you've got a beautiful child, they have a rare disease, we have a plan, we have a medicine, we have a gene therapy that could provide great benefits and hopefully a cure. So that's the hope that we need to deliver on. So thank you all. This was terrific for your thoughts and most especially for just all the great work you do. You guys deliver real hope. Thank you.